Today we're going to talk about the rapid development of genetics in the early 19th centuries. And I just remind you that we talk about the seal theory in the previous class. A few classes before, we talk about the discovery of Mendel, and also we talk about the work of Thomas Morgan locating the genes on the chromosomes, and these and our discoveries led to a rapid development in genetics, and we're going to talk about a few of those discoveries. We're going to focus on the discovery of the DNA. We're also going to talk a little bit about stainings and DNA and how they found that chromosomes are very important and are related to DNA. We're also talking about the transformation principles. And then we're going to talk about Avery experiments, how they found that DNA is the transformation principle, and we're using a very similar experiment, how they complement, how Avery complement those experiments. And finally, we are going to finish talking about Hershey and Chase experiments and how they found the DNA is the genetic material of T2 fakes. Later at the end, you can watch a small documentary and answer some questions about this that we will discuss more in detail, or how Watson and Crick found the structure of DNA, and you can answer a few questions about it. In the previous classes, we talked about three main scientific discoveries. In the last class, we talked about that the continual improvement made to microscopes over time led to the discovery of cells in the 17th century, and after almost 2 centuries later, the cell theory was formulated by Schwamm and Sladen. We also talk about the importance of the discovery of Mendel, that several scientists independently found similar results to those found by Mendel, and in particular, we focus on those found by Hugo de Vries. Finally, we talk about that by 1915, the basic principles of Mendelian genetics have been studied in a wide variety of organisms, and most important, or most notably, uh, the work of Thomas Morgan using fruit fly was very important because he found that genes were located on chromosomes, and the chromosomes would play a very important role in heredity. In the early 20th century, the identification of molecules of inheritance loomed as the major challenges to biologists. In this slide, I'm just going to show you that after the rediscovery of Mendel, in there was a rapid growth in genetics, and we are just going to focus on 4 experiments, but there were many more that led to the rapid development of genetics. Our second topic of this class, we will study the DNA discovery by Frederick Mesher in 1869, and possibly now the discovery of the DNA, we consider that is a huge discovery. However, at that time, it was not very important. Actually, this scientist died without knowledge, without knowing the importance of his finding. Why? Because at that time, proteins rather than DNA were considered uh, the carriers of the hereditary information, and I will show you that for multiple decades, despite this discovery, most of the scientists continue to believe that protein, not the DNA, were the key molecule in charge or involved in the inheritance. So a little bit of the life of this scientist. He was born in Switzerland and he wanted to be a priest, but his father suggested him to study, opposed to this idea, and suggested him to study, to go to med school. He went into med school and he joined a lab that he was working in trying to understand the components that cell has. So they knew that cells are composed of lipids, proteins, and and they wanted to know more and to study what other components the cells have. He had a task. The task was to study white blood cells, and this is why I in this diagram here. In this diagram, you can see that there is an infection. And why? Because white blood cells are in charge of protecting the body against diseases. So here you have a bacteria, and when you have a bacteria, white blood cells go to try to protect the body, and you find, and there is, in those sections of the body, you find pus. To extract white blood cells, it was very difficult. We have to extract it from lymph glands. On the other hand, from pus, it was very easy because there were a lot of white blood, and from there he extracted DNA. So in this slide, I would like to show you the experiment that the scientists performed, that the scientists conduct. The first thing that he did was he took bandages from a local hospital. Then he used these bandages to isolate DNA. And how he did it? By using a weak alkaline solution that allowed him to break the cells, to list the cells. And this, as a result of this, the nuclei precipitate out of the solution. Yeah, so he collect white blood cells from the past, then he lists the cell and isolate the nuclei, and the nuclei precipitated out of the solution. And as a result, he found a substance that he called nuclein that is present in every cell that he tested. And he found something very interesting, that it has a high concentration of phosphorus. Then he moved to another experiment, tried to test where in other type of cells also have this DNA, these nucleic acids, and he used salmon from sperm from salmon, and he analyzed that and found that he also found DNA. So again, he didn't know the structure of the DNA, he only knew the components. He only knew that it has the following elements, so he could have a hundreds of configuration. This double helix come much later. And now let's talk about the present, how we isolate DNA right now. DNA isolation is a very easy technique, and maybe in high school you were able to isolate DNA. It's a process of purification of DNA from samples using a combination of physical and chemical methods. So if you can go to YouTube and you can find a video of how to extract DNA from strawberries. You just need to use detergent that it will break the membrane, spoon of salt, water, and alcohol, and you can get a structure like this, all the DNA that it was found in the strawberry. And why I wanted to show you that, because similar to sewing thread, the DNA is a linear structure that it will tangle very easily. So in future slides, I will show you the importance of chromosomes, that DNA has to be very organized, wrap around proteins called histones, because it's a very long structure that we need to protect very well. And maybe you have heard about how long the DNA is. For example, you have here that for the total length of all DNA strands in a human body is equal 60 times the distance between the moon from the earth. And these are the calculations that they show. However, what I want that you remember is that the DNA need to be very well packed, so the information is protected from one generation to the other one. And I will show you how histones and proteins are fundamental to properly pack the DNA, and how chromosomes are able to highly coil the DNA to move it during cell division. Now, in the third section of this class, we're going to talk about the relationship between DNA and chromosomes. And to do that, we're going to show you how chromosomes were stained. So chromosomes come from the Greek root chromo, that mean color, and some that mean body, so colored bodies. And the first one who observed a chromosome was Carl Wilhelm von Neely, that he studied those chromosomes. He observed chromosomes in plant cells. And Walter Fleming, he studied in a lot of detail chromosomes during mitosis. He was born in Germany, and he was a military physician during the Franco-Prussian War. And he observed chromosomes by staining them with aniline dyes. Yeah, aniline dyes are a byproduct of coal tar, and he used them to study big cells of developing salamander embryos. And using those chromosomes, using those cells, he was able to see very clear chromosome, and he followed how this chromosome moves during mitosis. While biologists become convinced that chromosomes were physical seeds of genetics inheritance, chemists were claiming that those structures were made of both proteins and DNA. So, as I said before, there was a big debate between proteins and DNA. Many scientists of the day actually thought that it was proteins, the one in charge of carrying the hereditary material, because there were 20 amino acids for building a protein that produce a polymer, while DNA polymer are made only of 4 nucleotides. And amino acids were much more diverse. They can interact and combine in multiple ways, and they are actually very dynamic, and they can interact with multiple molecules. They can form bones and different type of tissues, while DNA was very unpopular. Nobody knows them very well, and they considered that it was a very simple molecule. And they saw that it was impossible that all the diversity of life was composed by such a simple molecule. So the genetic material was unconvincing because a crude chemical analysis suggests that DNA lack the chemical diversity needed in a genetic substance. Okay, now what we know about chromosomes in the present. So here in this diagram, I just wanted to show you how we go from the to a very small scale where you can see only the double helix, here to a big scale where you see the chromosome. So in the small scale, you will see the DNA that is wrapped around these structures called nucleosomes, that are the proteins that are formed by histones that I will talk in a second. And if you see all these histones comes together, and they give a tightly packed organization to the DNA. So we know that in eukaryotic cells, in the nucleus cell, the DNA molecule is packed into a thread-like structure called chromosomes, and we know that each chromosome is made of DNA tightly coupled, many times around by proteins called histones that support this structure. As I showed you before, the histones will be here. These are the nucleosomes, and I just tried to give you a mental picture how the DNA wrap around using the SUN thread. Actually, it looks like that. Each of these blue structures are called histones, and a nucleosome is the basic structural unit of DNA package in eukaryotes. Nucleosome core particles consist of 146 base pairs of DNA, and these structures inside, as I said before, there are different type of histone. You have the histone H2A, H2B, and here this one is called H21. So to finish, the chromosomes were formed by DNA and proteins. However, the one in charge of carrying the information was the DNA. The proteins over there were in charge of tightly packing the DNA. Now, to summarize all the information that we have seen, I'm going to show you a video that it will talk about these key points that I just explained.
This body, what is it? How did it begin?
The discovery of DNA itself as the hereditary material was not the discovery of Watson and Crick.
About the time that Darwin published The Origin of Species, a Swiss-German scientist named Friedrich Miescher was looking for a project. And his advisor said, see if you can find out anything about the nucleus. Miescher went down to the local hospital and got some bandages from patients and scraped off the pus, because that was a great source of white blood cells, and each one of those has a nucleus. So he had a little vial of cell nuclei, ground them up, and did chemical tests on them. And he discovered in the cell nucleus this new material. He didn't know what it was. He discovered DNA. But DNA is not yet thought of as the material of the genes. It's still pus on a bandage. We knew by about 1910 that genes were associated with chromosomes. Chromosomes were known to be made of DNA and protein. It wasn't clear which one was the hereditary material. Protein is most of the interesting stuff that goes on in your body. Tissue, muscle, blood, sinew, organs. Around the turn of the century, a number of people studied the biochemistry of DNA, found out that it has these 4 subunits. Protein has 20 or more different building blocks. So it seemed obvious that protein was a more complicated molecule, so probably it was the material of the gene.